Hello dear students. In this lecture we will discuss Agrobacterium mediated transformation. Agrobacterium tumefaciens and Agrobacterium rhizogenus are soil bacteria which causes crown gall disease and hairy root disease at wound sites on dicotyledonous plants. And once these tumorous growth they are formed, they can continue in the absence of these bacterium, and these tumor tissue can grow without the supplementation of any hormone on tissue culture media. So these tumor tissues, when they are formed, they synthesize some amino acids and sugar derivatives which are called as opines, and the type of opine which is synthesized in the tumor tissue, it is dependent on the agrobacterium strain that initiated that tumor. So there are two types of opines, octopine and nopeline, and these are derived from arginine, and they are very easy to detect in the tumor. So as you can see here in the picture, there is a tumorous growth on the stem which is called crown gall, and it is caused by Agrobacterium tumefaciens. And in the bottom picture you can see that the hairy roots are formed which are caused by Agrobacterium rhizogenus, and these are due to the presence of a special kind of plasmid present in these bacteria, and these tumors and hairy roots are caused by Ti plasmid, which is present in Agrobacterium tumefaciens, and Ri plasmid, which is present in Agrobacterium rhizogenus. So as you can see here, that this is a crown gall disease which is caused by Agrobacterium tumefaciens, and we will be focusing on transformation by Agrobacterium tumefaciens in this lecture. So you can see here that the plasmid is present in Agrobacterium tumefaciens, and this plasmid is causing this crown gall, and the plasmid is called Ti plasmid because it induces tumor once it gets into plant genome. A special part of Ti plasmid, that is tDNA region, goes and gets integrated in the host plant. So Agrobacterium tumefaciens, it causes tumors which are called crown gall in many plants when the bacteria infect a wounded site of the plant, which is usually on the stem which is close to ground. And uh, when this wounding takes place, the portion of Ti plasmid, that is tDNA, this is cut and it gets integrated into the genome of the host plant by some unknown mechanisms. So this uh, tDNA, it encodes the enzymes for auxin and cytokinin as well as opines when it is transferred to the host genome and when it gets integrated in the host genome. And so because of this, the proliferation of the tissues takes place and leads to the formation of tumor. Now these hormone synthesizing genes, they are known as oncogenes, and whenever the artificial transformation has to be done using Agrobacterium, genetically engineered Ti plasmids are formed in which the oncogenes are replaced with the gene of interest. It means that the oncogenes are removed, and then it is called disarming. So, for the Agrobacterium to be an effective vehicle for DNA transfer, the tumor causing genes of Ti plasmid are removed, and it is called disarming. These are replaced by our foreign gene of interest, and after disarming, the Agrobacterium tumefaciens is unable to cause tumor. So, in the right hand text, you can see that there is host plant and there is Agrobacterium, which is disarmed. So, when the host plant is wounded, it releases a specific chemical, which is acetosyringeon, which comes out of the wounded cells of host plant. Now, the acetosyringeon, it attracts Agrobacterium, which is growing in the soil, and in the next step, the Vir genes, which are also present on the TI plasmid. They encodes and restriction endonucleases, and these restriction endonucleases cut the tDNA part, and by some unknown mechanisms, this tDNA moves and gets integrated into the host genome, and this is how a transgenic is developed. So, this is in general the mechanism of how the uh, Agrobacterium causes crown gall in plants, and that is why this is called Agrobacterium tumefaciens is also called natural genetic engineer. So now let us see the detailed structure of TI plasmid. So TI plasmid, which is found in the virulent strains of Agrobacterium tumefaciens, is around 200 to 250 kilobases in size, and they have basically four regions: tDNA, that is the first region, which is called transfer DNA; Vir region, that is the second region, it is called virulence region; third region is con region, which is conjugation region; fourth region is ori region, that is origin of replication. So the first two regions, tDNA and Vir, they are associated with the tumor formation, and third and fourth regions, con and ori, they are associated with conjugative transfer and replicative maintenance of plasmid within the agrobacterium. So this is the detailed structure of the TI plasmid of Agrobacterium tumefaciens. You can see in the picture, which is on the left hand side, that TI plasmid has tDNA region, and this tDNA region, this is delimited by left border and right border, which has direct repeats, and this tDNA region has genes for auxins, auxin and cytokinin synthesis, as well as opines. And along with the tDNA region in TI plasmid, you can see that there is a virulence region and there is a origin of replication, as well as opine catabolism uh, region. So in the picture, uh, these are the two pictures where you see and that structure of TI plasmid. On the right hand side of the picture, you can see that the virulence region has many genes present, which are called as Vir A, Vir B, Vir G, C, D, E, F, H. So they have different roles to play during transformation when the tDNA gets transferred to the host genome, that we will discuss. So we have just discussed that the major regions of TI plasmid of Agrobacterium tumefaciens, what are their roles. Origin of replication, ORI, is responsible for the replication of TI plasmid, and virulence region, which is called Vir region. This region has genes called Vir genes, whose products enable the transfer of tDNA from bacterium to plant cells. Then the tDNA region, this is a region of TI plasmid which contains the oncogenes, and it is flanked by 25 base pair direct repeats on both the sides, which are called left border and right border. And the genes which are present in this region are IAAM and IAAH genes, which are responsible for the synthesis of auxins, and cytokinins. And the fourth region is for opine catabolism. And we have another region which is a region for conjugation also present in Agrobacterium. So during tumor formation, a defined sequence of Ti plasmid, that is your tDNA, is transferred to the plant cell and it integrates in the nuclear genome. So one or more copies of the tDNA can be present in the plant cell. And we have just discussed that tDNA in nopaline strains of Agrobacterium tumefaciens, the tDNA region is around 24 kilobases, and in octopine strains, the two segments of tDNA, which are TL and R, they are TL is 14 kilobases, is found in all transformed lines, while TR is 7 kilobases. It is not found in all tumor lines. So you can see that at the picture at the bottom is showing the structure of tDNA, which is your nopaline kind, and in tDNA region you can see the genes responsible for auxins and cytokinin synthesis. So here you can see that the tDNA region has genes which are called TMS2, TMS1, TMR. These are responsible for the synthesis of cytokinins and auxins, and this is a region which is present in tDNA. This is octopine DNA. You can see that these are the details of octopine tDNA region, and we have just discussed that in the mechanism of tDNA transfer, the wounded host cell, they release acetylcysteine, and because of which the agrobacterium is attracted. This is called chemotaxis, and on upon it and forms a complex with host cells, and then Vir genes are activated. And when the Vir genes are present in the virulence regions, they have different role to play. Vir A is the chemoreceptor of acetylcysteine, and it makes a complex with acetylcysteine and activates Vir G. Then Vir B, C, their roles are unknown, but probably they have a role in extracellular transport of tDNA. Vir D encodes restriction endonuclease. Vir E helps the tDNA to integrate in the plant genome, and Vir G activates rest of the Vir genes. So now there are two kinds of vectors in Agrobacterium tumefaciens. Cis vector, which is called co-integrate vector. Here the tDNA and Vir genes are present on the same plasmid, and the other kind of vector, it is called trans vector or binary vector, which are also called shuttle vectors. Here the tDNA and Vir genes are present on separate plasmids. So in co-integrate vectors, which are called cis vectors, you can see that the tDNA which is present in Ti plasmid, it is combined with the intermediate vector in which the foreign gene and marker gene like reporter and selectable marker gene are there along with our foreign gene of interest. So this becomes a co-integrate vector or cis vector which can replicate only in the agrobacterium, and the co-integrate vector has both the Vir gene and the tDNA on it. The other vector is your trans vector, which is also called binary vector. So here the two kinds of plasmids are present. First is called helper plasmid, which has only Vir region, and the other has binary vector, which has the tDNA part. So the binary vector can replicate in both the E. coli and Agrobacterium, and this is called transvector. So, students, we will discuss the second part of this lecture where we will discuss how the genetic engineering of TI plasmid takes place for genetic transformation of crop plants. So, thank you, students. See you in next lecture.